Good morning. Let's discuss on vitamin B12 or cobalamin. It is a water-soluble vitamin which is important for development of central nervous system, including brain and spinal cord, and it's also important for RBC's development maturation. So structurally, vitamin B12 has a corin ring containing the pyrrole similar to the porphyrin ring, linked to the cobalt atom held in the center of the corin ring by four coordination bonds with the nitrogen of the pyrrole. So these are the four pyrrole rings, and their nitrogens are coordinating with the cobalt in the center, and remaining coordination valencies are satisfied by combining with one nitrogen from the dimethyl benz-imidazole nucleotide. So one attachment is through this nitrogen, and the sixth one, sixth bond is linked to either methyl or 5 deoxyadenosyl or hydroxy group to form the methyl cobalamin or uh, the adenosyl cobalamin. So this is the fifth valency. R can be a methyl group or it can be adenosyl group. So this is the basic structure of cobalamin. So, what is the RDA or requirement? Daily requirement is 1 to 2 micrograms per day. During pregnancy and lactation, this amount increases to 2 micrograms per day. Active form of vitamin B12 is methylcobalamin and deoxyadenosyl cobalamin. What are the sources? Dietary sources of vitamin B12 are mostly of animal origin. They are meat, egg, milk, dairy products, fish, poultry. Vitamin B12 is absent in plant food. Human obtain a small amount of B12 from the intestinal flora as well. And curd is a very good source because lactobacillus can also synthesize vitamin B12. Let's see the digestion absorption of vitamin B12. So, because vitamin B12 is complexed with the food products, so once in the mouth, the salivary glands secrete the saliva. Along with the saliva, there is a secretion of cobalafillins. It is a kind of protein which binds with vitamin B12 and protects it from the dangerous effect of HCl in the stomach. So all these products, vitamin B12, food products, and cobalamin finally reach the stomach. In the stomach, there is a stimulus to the parietal cell to secrete the intrinsic factor from the parietal cells and HCl. So what happened? B12 is actually the extrinsic factor because the factor which we derive from the external sources. An intrinsic factor is secreted by the gastric parietal cell. So this your intrinsic factor is actually a glycoprotein with a molecular weight of around 50,000. So what happened in the stomach under the influence of gastric proteases? There is a no melting of food product or digestion of food product, and there is a freeing of vitamin B12. Now B12 winds up with cobalafillins, so that cobalafillin prevent the vitamin B12 exposure to the HCl. Otherwise, it will be damaged under the exposure of HCl. So finally, this combined vitamin B12 and cobalamin complex reach to the intestine. So, and the intrinsic factor of castle, it is intrinsic because it is being secreted from the parietal cell, and vitamin B12 is the extrinsic factor because it is being coming from the food, from the external sources. Finally, the food product reaches into the duodenum. So, in the duodenum, the cobalafillin is hydrolyzed by trypsin. It is being hydrolyzed by trypsin and of the pancreatic juice, and the vitamin is finally released up. It is freed up, and then vitamin binds to intrinsic factor. Now here in the small intestine there is a binding of intrinsic factor to vitamin B12, and this complex is now um, um, it is may not be, in pancreatic insufficiency, the vitamin may not be released. So then vitamin cobalamin complex is excreted and resulting in vitamin deficiency. But in normal condition, what happens? The one molecule of intrinsic factor binds with the two molecules of vitamin B12, and this complex is attached with the specific receptors onto the mucosal cell, especially in the ileum, and through these receptors in the ileum, it is being internalized, and finally it reaches into the portal vein. So it may be noted that vitamin B12 is basically absorbed from the ileum, while the folic acid is being absorbed from the duodenum. Finally, once in the portal circulation, so it is all set to wind it with the transcobalamin 2 protein. So in this complex form, it is now transported to the liver, where it is transcobalamin is actually a glycoprotein. It is a specific carrier and it is stored in the liver cells. So the adenosine B12 form in combination with the transcorin. So that used to be stored in in few microgram quantities in the liver. And B complex vitamins are not stored in the body generally, but B12 is an exception because there is a stay of this vitamin in the liver, so which is sufficient for the requirement for 2-3 years. So vitamin B12 deficiency is seen only years after the gastrectomy. Understood? So then move on to the so storage is actually in few milligram, 2-3 milligrams quantity. So now move on to the biochemical functions of vitamin B12. There are only two human enzyme systems that are known to require vitamin B12 coenzyme. One is the isomerization of methyl malonyl coenzyme A to succinic acid. This reaction already we have seen. Then conversion of homocysteine to methionine. So we learned about this in the methionine metabolism. So this is a schematic presentation of the how it's being absorbed and transported. So peptic digestion releases the vitamin B12 in the stomach. So vitamin B12 now bound to the heptocorrin or cobalamin. It sounds the same. So heptocorrin B complex, B12 complex digested in the duodenum under the influence of trypsin. Then finally, the released vitamin B12 is all set to wind up with the intrinsic factor of Castle, and vitamin B12 is an extrinsic factor. So both these factors are complexed. Finally, they reaches to the ileum. In the ileum, this complex binds to the cubulin. Cubulin is a type of intrinsic factor receptors. So they get internalized through these receptors. This complex is internalized and absorbed complexes with transcobalamin 2, and finally it is transported to the liver via portal vein. So we'll see here the role of vitamin B12 in isomerization of methyl malonyl coenzyme A to succinyl coenzyme A. So valine, methionine, isoleucine, and odd chain carbon fatty acid. They all lead to formation of propionyl coenzyme A as a metabolic product. So it in the presence of biotin and vitamin and the propionyl coenzyme A carboxylase, it converts up into the D-methyl malonyl coenzyme A. Actually, propylyl coenzyme is a useless product which is finally converted into very, very useful product, that is succinyl coenzyme A, which is at one side important component into intermediate of TCA cycle, while at another side it is one of the important component or precursor for synthesis of heme. So from propionyl to D-methyl malonyl coenzyme A, finally in the presence of enzyme D-methyl malonyl coenzyme A racemase, it converts up into the L-methyl malonyl coenzyme A. So here comes the role of vitamin B12 in the form of deoxyadenosylcobalamin cobalamin. So it influences this enzyme methyl malonyl coenzyme isomerase or mutase. So it is a coenzyme for this enzyme. So the succinyl, finally, L-methylmalonyl coenzyme A converts up into the succinyl coenzyme A, and which is now involved in the TCA cycle as well as for synthesis of heme. So if this vitamin B12 is lacking or absent or deficient, so it starts, this L-methylmalonyl coenzyme A starts appearing in the urine. Now, another important role of vitamin B12 is in conversion of homocysteine to methionine. It requires the coordination of folic acid as well. We have previously seen in this reaction in folic acid metabolism. So homocysteine to methionine, this just one methyl group difference is there in between these two products. So the enzyme is methionine synthase, which is dependent on vitamin B12. So cobalamin to methyl cobalamin. And this methyl cobalamin, how it is being formed, it requires the methyl tetrahydrofolate. So, which in the presence of vitamin B12, it converts up into the tetrahydrofolate and gives it methyl group to cobalamin. This is how there is a generation of methyl cobalamin, very, very important inducer of this or coenzyme of this methionine synthase. If there is a deficiency of this enzyme, there will be a folate trap because this is an irreversible reaction. Methyl will remain attached to the THF or tetrahydrofolate, and in this is how the tetrahydrofolate is entrapped along with the methyl group. So homocysteine, now its uptake, the methyl group converts up into the methionine. So we can nicely see what is how important role plays vitamin B12 and folic acid in conversion of homocysteine to methionine. There is more clear vision of important role of vitamin B12 and folic acid. We have just seen vitamin B12 and folic acid needed for conversion of homocysteine to methionine, and it is also important for synthesis of thymidine triphosphate because this combination, important for conversion of UMP to TMP or thymine monophosphate, which is important nucleotide for synthesis of DNA. So then this TTP is important for synthesis of DNA. So this is how, if the reaction is hampered or folic acid or vitamin B12 is deficient, so DNA synthesis will be hampered. Then how the deficiency will affect the RBC's maturation. So impaired DNA synthesis, there is a delayed cell division as well, and hemoglobin synthesis is while it is normal. So in such condition, the defective nuclear maturation will be there. It will lead to the formation of megaloblastic anemia. So immature, large sized with the large nucleus. These are the RBCs, and they are now in the general circulation. So this is macrocytic anemia or megaloblastic anemia. And these giant cells are also known as giant metamyelocytes. This is all due to the deficiency of either vitamin B12 or folic acid. So deficiency manifestations are in the form of pernicious anemia, megaloblastic anemia, methylmalonyl aciduria, then neuropathy, subacute combined degeneration, and folate trap. So pernicious is because of no deficiency of intrinsic factor. If his intrinsic factor is not there, so there won't be proper absorption of vitamin B12, and that's how there is a deficiency of vitamin B12. Then neuropathy is in a very advanced form. This is known as subacute combined degeneration, and folate trap is there. Then causes, what are the causes for B12 deficiency? So nutrition is the one cause, lack of proper nutrition. Only the source of vitamin B12 in vegetarian diet is curd or milk. Then decreased absorption, gastrectomy or resection of ileum and malabsorption syndrome. So all these lead to decreased absorption. Then additions, pernicious anemia, antibodies are generated against the intrinsic factor, so becomes deficient, leading to the defective absorption of B12. Then gastric atrophy, in case of gastric mucosal atrophy, it happens. Then during pregnancy, increased requirement of vitamin in the pregnancy. And there is one condition, fish tapeworm. It is seen actually in the Scandinavian countries. This tapeworm actually it is a parasite and it has a special affinity to vitamin B12, causing the reduction in the concentration of this vitamin in the body. Then these are the various manifestations. Once more, there's a clear account of these disorders. Folate trap, vitamin B12 deficiency causes the simultaneous folate deficiency due to the folate trap. It remains in the form of methyl THF. Then megaloblastic anemia in the peripheral blood, megaloblasts or immature nucleated RBCs and hypersegmented neutrophils are observed. So you can see the hypersegmented neutrophils are there. Then immature RBCs, large size megaloblastic cells are there. Then abnormal homocysteine level due to the folate trap. There is no conversion of homocysteine to methionine, so it remain increased in the blood, and from blood it is causing the homocystinuria as well. And the homocysteine level in blood is associated with myocardial infarction. Then demyelination, very, very important manifestation due to the deficiency of this vitamin, as active methionine is not available in the form of S-adenosyl methionine. So methylation of phosphate ethylenol to phosphate choline is not adequate because this also requires SAM, and this leads to the deficient formation of myelin sheaths of nerve, demyelination, and neurological lesions. So this is the picture of peripheral blood picture of megaloblastic anemia in vitamin B12 deficiency. Normoblast, they are immature nucleated RBCs and their hemoglobin remains normal and hypersegmented neutrophils are seen. Then vitamin B12 deficiency also causes the parallel ridges in the nails. What is subacute combined degeneration? Actually, it causes damage to the nerve, and there is a demyelination affecting the dorsal column and pyramidal tract of spinal cord. So both these are simultaneously affected, dorsal column as well as the pyramidal tract of spinal cord. So that's how it is being named the combined degeneration. Then symmetrical paresthesia of the extremities, alterations of tendons and deep sense and reflexes, loss of position sense, unsteadiness in the gait. Then positive Romberg sign. What is a positive Romberg sign? Here, if a patient is asked to stand with the heels together, first with the eyes open and then with eyes closed. So while closing the eyes, if he falls down, so it is a positive Romberg sign. Then he also shows, patient also shows the positive Babinski sign. That is an extensor plantar reflex. So, normal plantar response is there when we are, touching the extensor plantar reflex. We are testing the extensor plantar reflex. So, in the normal condition, there is a downward movement of toes and there is a flexion of all other toes. But if the patient is having the deficiency of vitamin B12, so this will lead to fanning of other toes along, and the big toe is moving upwards. So this is a positive Babinski sign. Then achlorhydria, like absence of acids in the gastric juice, is associated with vitamin B12 deficiency. I have already showed you or told you about like how it is involved in freeing of this vitamin B12 while it is complex with the food products. So the vitamin B12 deficiency is manifested in the nails as a parallel ridge. Then serum B12 level, how we will diagnose the deficiency. First is estimation of serum B12 level by ELISA or RIA method. Then a peripheral smear picture. Peripheral blood and bone marrow aspiration shows typical picture of megaloblastic anemia. Third is the Schilling's test. In this particular test, the radioactive labeled vitamin B12, 1 microgram is given orally, and simultaneously the intramuscular injections of unlabeled vitamin B12 is given in order to saturate the tissues with normal vitamin B12. So the radioactive vitamin B12 will not bind to body tissues. Therefore, in normal person, the entire absorbed radioactivity will pass into the urine. But if the patient is suffering from pernicious anemia, so less than 5% of radioactivity is detected in the urine. Then methyl malonic acid test in the urine is also such test to diagnose the deficiency. Then forma-aminoglutamic acid, FIGLU conversion of FIGLU to L-glutamate is mediated by folic acid, and vitamin B12 also has a role. So will if the test is positive, there is a deficiency, then homocystinuria is another indication for the deficiency of vitamin B12. Treatment lies like all macrocytic anemias are generally treated with folate and vitamin B12 combined. So combined therapeutic dose of B12 and folic acids are given by to the patient.